Okay, are there any questions before we start? So we're going to be moving on to the urinary system, and then we're going to do reproduction, and then we're going to do pregnancy and development, and then a little bit of genetics. And so that's kind of the end of the whole class.
That's crazy.
Yeah, I know. We're almost yeah. Okay, so when we talk about the urinary system we know produces urine, but it actually has some other functions. And one function is that it helps to regulate water solute balance in the body. Okay, so remember that is important because of the way that things move, right, via osmosis, and that it is also important to regulate solute concentrations because we don't want them to get too concentrated or too dilute as you take in water from the environment. So like drinking water. So I don't know if you realize this, but or you might have heard of this, but when people have contests where they see how much water a person can drink, that is actually really dangerous because you can actually die from taking in too much water, right? So you don't want to drink so much water that your kidney is not able to regulate the concentration of the water in the solutes and get rid of that excess water, okay? So it regulates water. It also regulates ion concentrations in interstitial fluid. So the interstitial fluid is the fluid that is around each and every one of your cells. And when we talk about ions, we can talk about sodium and chloride and also potassium. And the book mentions that potassium is really important. And so when we look at the function of the kidneys are actually able to reabsorb ions depending upon if they're needed by the body. So we can talk about the reabsorption of certain ions in the body. We also have to get rid of nitrogenous waste. So this is the excretion of nitrogenous wastes. Right? So ammonia, which is the breakdown, right, of proteins, can be converted into a, and that's not how you spell ammonia, I think there's two M's in there, ammonia. Gets converted to urea. And so urea is our nitrogenous waste that we use. Sometimes we can get uric acid also forming in the body, and uric acid crystallizing can sometimes be a problem. So for example, in people that have gout, they get excessive uric acid in certain joints, and that can be very painful, right? But we excrete our nitrogenous waste as urea. Okay, the other thing that it does is it regulates acid-base balance. So we talked about how the respiratory system also helps to regulate acid-base balance. So we talked about respiratory acidosis and respiratory alkalosis, but the renal system also regulates specifically like blood pH, right? So it regulates blood pH. And the reason for this is that sometimes our urine is very acidic and other times it's not so acidic. And so if you have excess acid in your body, then you can actually urinate it out, right? And the urine, sometimes urine burns the skin if it gets on the skin, and that's because it's really acidic, right? So the urine can be, so the pH of the urine varies. Right? And it varies because if we're needing to absorb the hydrogen, reabsorb the hydrogen ions, we do. If we need to get rid of excess hydrogen ions, we get rid of it in the urine, okay? It also regulates or helps to regulate blood pressure. So when we talked about the endocrine system, we specifically talked about aldosterone. And does anybody know or remember what endocrine gland secretes aldosterone? It's the one that's right by the kidney. Adrenal glands, right? And so this is said to be a mineral corticoid. Hormone. And so it's secreted by the cortex of the adrenal gland. Remember that the medulla of the adrenal gland is actually nervous tissue. And it's a mineral corticoid hormone. And it increases the absorption, reabsorption of sodium by the kidneys. And we'll talk about how that reabsorption occurs in a little bit, okay? And then when sodium is reabsorbed, water passively follows. So water is passively reabsorbed. Right? So how is that going to affect blood volume if aldosterone is present? Is that going to increase or decrease blood volume? If water is passively reabsorbed from the filtrate from the kidneys, then it would increase, right? So this is going to increase blood volume. Now the kidneys also produce another chemical that also influences blood pressure, and this chemical is renin. So it produces renin. So the kidneys, we'll put this one. So the kidneys, this is still under regulating blood pressure. Kidneys produce renin. And remember that renin causes angiotensinogen. So I'll just put angiotensinogen. So remember when we were talking about pepsinogen? This is the inactive form, right? So it can get converted to angiotensin II. Right? And this is renin. So renin does that. Right? Inogen means inactive. So like pepsinogen, chymotrypsin, trypsinogen, right? Some of those words that we learned when we were talking about enzymes in the digestive tract. So this angiotensin is a vasoconstrictor. Right? So vasoconstriction causes there to be the smooth muscle, circular smooth muscle contracts around the blood vessels, and it causes the diameter of the blood vessel to become smaller, and so this boosts blood pressure. So this would increase blood pressure. Okay. Now we also learned that the kidneys produce a hormone that regulates red blood cell production, and what is that? EPO. So the kidney produces EPO. So you should remember that EPO, right, is erythropoietin, which is a hormone that stimulates the bone marrow, the red bone marrow, to undergo erythropoiesis, which is the production of red blood cells. Okay. The last one, which actually I didn't even know until I read your book, which is that the kidney is capable of gluconeogenesis. Okay, so maybe you heard about this in, if you took nutrition. How many people, have you guys all taken nutrition? Yeah. So gluconeogenesis means that you're producing glucose from a non-carbohydrate precursor. And so glucose is so important, right, specifically for the nervous tissue. It just runs on glucose. That if you don't take in enough carbohydrates, then you can start converting proteins and lipids into glucose. And so this is the production of glucose from a non-carbohydrate molecule. And the liver does this, but also the kidney does this. So this is the production of glucose from I'm running out of room here, non-carbohydrate. Right? So protein or lipid. And so you could say that the kidney, therefore, helps to regulate glucose levels in the blood. So if you're fasting, or maybe you're starving, or you're in a state of famine, then the kidney plays a role in regulating blood glucose levels, which is kind of interesting because, you know, the kidney is one of the first things that people have problems with, it seems, when they start to get diabetes, right? Type 2 diabetes, which is the inability to regulate blood glucose levels. Okay, so those are all the things that the kidneys does. So let's look at a picture of them. So this large blood vessel coming down, actually it would be going up, is, what do you think is that? It's a vena cava, and would it be superior or inferior? Inferior. So then we have our inferior vena cava. And then we have this large blood vessel, and that would be, what do you think? Yep, so this is actually the aorta. So this is the descending aorta, or you could sometimes call it the abdominal aorta. Okay. And so the branches off of these that go to the kidney, actually, in your book it says that 1/4 of all the blood that's pumped out of the heart goes to the kidney. So the 1/4 of the cardiac output. So that's a huge amount, like it's 1.2 liters for every heartbeat, right? So it's a lot of blood that's going to the kidneys. So this blue vessel right here, this is actually called the renal vein. I know yours is not blue, but it would be the one that's most is on top, okay? And then underneath that, the red one is the renal artery. So the kidney plays a huge role in filtering the blood, right? And by filtering, then it has the ability to regulate the body fluids as well. Okay, so what do you think these yellow things coming down from the kidneys and going to the bladder would be? Does anybody know? They're part of the urinary system. They're actually called ureters. And so you don't want to get that confused. This is a female, and so the ureters, then from the bladder, this would be the urethra, right? So the urethra is below the bladder. So this would be, I think they're showing the bladder here. So this is the bladder. And then this in the female is the urethra. It's also the male, but the urethra in the male travels through the penis. Okay. And then what is this up on top of the kidneys? These are the adrenal glands. Now, one of the really interesting things about the kidney is that they don't actually sit in the body cavity with the digestive organs. They sit actually behind the body cavity wall, or the lining of the body cavity, and so they're actually said to be in a retroperitoneal position. And so this is the kidneys. This is a cross-section of the abdominal area. Right, this is a cross section, and so this is the cavity within all our organs sit in. And so, what is that called? What is that big peritoneal cavity? Okay, so when we look at the kidneys are retroperitoneal. So they're behind the peritoneal cavity. Okay. So you can see the veins, and you can see the arteries here as well in the cross section. And you can see the vena cava and the aorta. One of the interesting things about the kidneys is that they're actually cushioned by fat. And so this fat is called the perirenal fat capsule. Okay, so this actually has a couple of functions. One is it cushions. So, you know, when you get kicked, or I don't know this, but if you get kicked in the back of your body, right, kind of up here by your, if you look at where this is, it's kind of where your iliac crest is, just a little bit above that. You could actually have damage to the kidneys, right? So if you have crushing injuries, like a car accident, or for example, you could have blood showing up in the urine because you damaged your kidney, okay? Now, the other thing that it does is it maintains its position. So it maintains the kidney position. And so in extreme cases, when people lose all of their body fat, say maybe they're anorexic or they've been going through a famine, the kidney can actually drop. And that is called renal ptosis. So renal ptosis is when the kidney drops. And when the kidney drops, because of the lack of fat, then it can kink the ureter. Right? So the ureter is generally straight, but if it falls, then you can get a kink in the ureter. And then what happens is that urine starts to back up into the kidney. And this has a name, it's kidney hydronephrosis. So this can cause urine to back up and can lead to hydronephrosis. So literally water, and we're going to learn about what nephros means, nephrons, but it's water on the kidney, right? And so this can actually cause kidney damage, and it can also cause the renal failure, right? So it can cause damage to the kidney, and then it can cause renal failure. So the kidneys just stop working. Okay. We also see that there is some connective tissue here, and that is what we refer to as the renal fascia. So this is the connective tissue, and it's on both sides. So this is the renal fascia. And remember that fascia is dense connective tissue. So it's a type of connective tissue. And then we also have on the surface of the kidney, we have what is called the fibrous capsule. So the fibrous capsule is on the surface. This is around the kidney. So it'd be like the visceral connective tissue on the surface of the kidney. Okay. Okay, so we're going to talk about the anatomy of the kidney. And when we look at the kidney and its shape, and so this is your diagram here, there's a shape to it where you have this indentation right here. And this is called the renal hilum. H-I-L-U-M, I think, or sometimes it's called the renal hilus. So that's where the blood vessels enter, and that is where the ureter leaves. And so I'm going to write this right here. This is the ureter. And then you can label the blood vessels if you like. Okay. We also have right here, and this is the renal hilus kind of enters into this area, and this is what is called the renal pelvis. And it is the renal pelvis that collects the urine. And then the ureter is what transports the urine to the bladder. Now, when we look at the histology, you can see that there is a difference between the outside and the inner part of the cross or the longitudinal section of the kidney. And so this part right here is referred to as the cortex. And this will be important because the reason why it looks this way and looks different than the medulla has to do with the types of tubules that are contained in those particular areas, okay? So that's the cortex, and then this part right here is what we refer to as the medulla. So the cortex is the outer part, and the medulla is the inner part. We also see that there's these weird shaped areas in the kidney, and these are what are called renal pyramids. So I'll say that this is a renal pyramid. There's more than one. And we'll look at why they look like this, why they look striated in a little bit. Okay, so that's the part of the kidney that you need to know for lecture. We might go into a little bit more detail in lab. Okay, so let's look at how the blood moves through and into the kidney. So I already mentioned that we have the renal artery. Okay. So the renal artery brings the blood in, and in your book, they have a lot of detail, and I'm just going to break this down for you in a little bit. So I'm going to say it goes into smaller and smaller branches here until it gets to what is referred to as the afferent arteriole. So remember the arterioles are just really small arteries. And remember that A comes before E, like even in homeostasis mechanisms, you have the afferent pathway and the efferent pathway, right? So it's a way to remember that the afferent pathway is the first arteriole, because we're going to go from the afferent arteriole to what is referred to as the glomerulus. And this is where filtration takes place. So the blood that doesn't get filtered out, the blood cells hopefully don't get into the filtrate, and lots of the other fluid doesn't actually get into the filtrate. It will leave via the efferent arteriole. Right? And then it actually branches into some other capillaries. And so the other capillaries include what are called peritubular capillaries, plus a special type of capillary bed, which we're going to talk about, called the vasa recta. Okay. And then it goes into bigger and bigger veins, right, until it leaves via the renal vein. Make that a capital, renal artery, renal vein, okay? So we're going to pay particular attention to how things get through that filtration membrane, the glomerulus, and also what regulates the blood pressure and the blood flow moving into the glomerulus. Because if you think about it, blood is coming under high pressure via the arterial system, and we have to protect our filtration membrane, because if the blood pressure is too high, we're going to damage the membrane. But if the blood pressure is too low, then we're not going to filter the blood properly. So we have to like regulate the blood pressures really well, specifically right here, in order to make sure that we get enough filtration being formed. Okay. So in your book, they have this in a little bit more detail. So from the renal artery, it goes into smaller and smaller and smaller until it eventually gets to the afferent arteriole, glomerulus, efferent, and then back up. So it just kind of goes through the entire kidney. Okay, so the nephrons. The nephrons are the functional unit of the kidney. And there's like 1 million, about 1 million in each kidney. And they are specialized tubules. So if we look at the first part of the nephron, we have the glomerulus. Now the glomerulus is a specialized fenestrated capillary. So that's kind of unusual. Fenestrated means that it has holes in it, right? So that means a lot of stuff can get through, but it also has specialized cells around the capillaries that are called podocytes. So fenestrated capillaries with podocytes. Okay, and this is where filtration takes place. So this is my filtration membrane. Oops, filtration. Okay, so we don't want big molecules like big proteins to get through, like albumin, for example. We don't want red blood cells or white blood cells to get through, and so that filtration membrane is very specialized. Okay, now we also have around the glomerulus what is called the Bowman's capsule. This can also just be called the renal capsule. So Bowman is just the name of the person that discovered it. And the glomerulus and the Bowman's capsule together make up a structure called the renal corpuscle. So the function, having a coughing fit, sorry. So those together make up the corpuscle. The Bowman's capsule functions to capture the filtrate. So this captures the filtrate. Okay. So let's take a look at the glomerulus. Okay. So this shows a diagram of the whole nephron structure that we're talking about, 1 million of these in each kidney, right? This is the capillary, so that's the glomerulus. Then you have the renal capsule or the glomerulus capsule, and this is also called the Bowman's capsule, right? And this is what picks up the filtrate, right? And then it goes into a series of tubules, okay? So from the renal corpuscle, it goes into what is called, so I'm just going to put, this is first part of the glomerulus, first part of the nephron is the glomerulus. The second part is the Bowman's capsule. The third part is called the proximal convoluted tubule, which you can call PCT, right? You don't want to have to write that out too many times. So proximal means that it's closest to the glomerulus. So it's nearest the glomerulus. Convoluted because it's highly folded, right? It has lots of folds in it. And this is next to what is called the loop of Henle. So after it goes through the proximal convoluted tubule, it goes into the loop of Henle, and then it goes into the distal convoluted tubule, which is abbreviated DCT in your book. And then finally, we get to the collecting ducts. Loop of Henle, Henle. That's a person's name. So you have the Bowman's capsule, and then you also have the loop of Henle. Yep. Okay. So you need to be able to identify these different pieces, right? On this one, it's really easy on this particular diagram because they kind of straightened out the tubules. So you can see proximal loop of Henle, distal convoluted tubule, and collecting duct. But if you look at them under what they really kind of look like, you're like, holy moly, right? It's like, it's so convoluted, right? So what you would have to do is you would have to make sure you were looking at, and maybe I wouldn't give this to you, but you'd have to make sure that you're looking at proximal is first right here, right? So it's coming out, and then the distal would actually be, do they have that distal? So this is, maybe this is proximal, and this is, oh, I can't even tell here. Maybe this is proximal. Anyway, so this is the way they really look, okay? So I give you the simplified version. Okay. Now notice how there are actually two different types of nephrons here. And so we want to look at those types of nephrons. So the first type is called the cortical nephron. And about 85% of our nephrons are of this type. So this is much more common. And actually, in most organisms, besides mammals, like fish and amphibians and lizards, all they have are cortical nephrons. So that is what is kind of ancestral, that type of nephron, okay? The other type of nephron is called, so there's some fun words in this lecture, is called the juxtamedullary nephron. And that's only about 15%. Okay. So this is a special adaptation for mammals, which means that with the use of the juxtamedullary nephron, this is the type of nephron that allows us to produce very concentrated urine. So this is the type of nephron that allows us to produce urine that is more concentrated than our body fluids. And desert mammals, like kangaroo rats, for example, have even more specialized, longer juxtamedullary nephrons. So if we look at what this means, juxta means next to the medulla. So the reason why they're called juxtamedullary is that their nephrons, or excuse me, their loops of Henle. So their loops of Henle extend down into the medulla. And so hence their name, juxtamedullary. So desert mammals tend to have longer loops of Henle, and they actually can produce more concentrated urine than we can. And that is the ability to conserve water, right? So this helps, I'll put conservation of water, right?
I wonder if that counts for people that live there too. Do they adapt?
I don't know. I don't think so. I think this is something that the nephrons are built when you're in embryo. Yeah. I don't know though. It seems like you might get better at it.
I mean, like people over time. Those,
yeah, it seems like you might. Oh yeah, over time. Yeah. Seems like you might be better at it if you go for long periods of not drinking water. Then it seems like you would acclimate to that, and your kidneys would get better at conserving water. I always wonder that because a friend of mine wants me to do this crazy thing where you don't drink for 4 days. I was like, how does that work? I don't know. Yeah. Okay, so juxtamedullary nephron. Okay, so this is the cortical. So notice the nephron does extend a little bit, or excuse me, the loop of Henle does extend a little bit, but this is the juxtamedullary. And it also has a specialized capillary bed around it that is called the vasa recta. Okay? So the vasa recta is around those long loops of Henle. So we'll write that. Oops, sorry. Okay, so let's talk about glomerular filtration a little bit. So when we look at the regulation of glomerular filtration, we can talk about GFR. So this is abbreviation for glomerular filtration rate. And this is one of the things that they measure or they try to estimate in people that are having kidney problems is like, okay, so what is their GFR, right? So are they sufficiently filtrating their blood, or is there an accumulation of toxins in the blood, and do we need to do something about that, right? So if we look at, let me get my notes, if we look at the typical glomerular filtration rate, it is about 120 to 125 milliliters per minute. Okay. If you have renal disease, so if it is less than, if your GFR is less than 60 millimeters per minute, then this means that you have renal disease. Okay. So one thing that affects GFR is the diameter of the afferent arteriole. So we can regulate the diameter of the afferent arteriole. Afferent arteriole, right? And this is actually called a myogenic mechanism. So what does myo mean? Muscle, okay? So what happens with arterioles is if there's lots of blood coming in, and maybe too much blood coming in, so let's say the flow of blood into glomerulus is too great, it could actually damage the capillary, then it's going to cause the arteriole to expand, and it will just contract in response to that, right? So this is a contraction in response to overdilation. And this protects the glomerulus. Okay. Now, some other things also affect the glomerulus, also affect arterioles and arteries. And one of them is a neural mechanism. And this is the sympathetic nervous system. And it specifically causes the release of epinephrine. Right? So epinephrine is going to cause what? Do you remember what epinephrine causes? Remember that EpiPen causes constriction, right? So this causes blood vessel constriction. So that's another thing that's going to affect the flow of blood into the glomerulus. So if we look at a diagram that shows this, this is my glomerulus. And you can see here that the red blood cells are not leaving, right? They're not going into the filtrate. So this is the afferent arteriole. So the membrane, which consists of those fenestrated capillaries plus these little podocytes. So these little yellow cells are the podocytes. So think cells with little legs on them, right? And those legs help to prevent big things from coming through, okay? And then we have the efferent arteriole here, right? So blood would exit in that direction, Okay. Now, there is another a mechanism for regulating the amount of blood that flows in, and it is due to what is called the juxtaglomerulus apparatus. Okay, so we're going to go back to that juxtaglomerulus apparatus. And the juxtaglomerulus apparatus, glomerulus, has specific cells that are able to detect stimuli. Apparatus. Let me spell this better. Glomerulus apparatus. Okay. So the first cells that we're going to talk about are the macula densa. And the macula densa are able to detect salt concentration inside the filtrate. And it actually is near the distal part of the nephron. So it's like at the very end when the filtrate should have already been modified. So salt should have already been absorbed or reabsorbed here. Okay. So if it is too salty, so if NaCl concentrations are too high, that means that filtration rate is too high. So filtration is too great. Okay. And the reason for this is that if you're producing too much filtrate and you're not able to pump and get rid of all the salt to reabsorb the salt, then there's going to be too much salt, right? So this is going to actually cause constriction of the afferent arteriole. So it constricts afferent arteriole and decreases GFR, the glomerulus filtration rate. Okay, so those would be like little chemical sensors, right? The macula densa are cells that are able to detect solute concentrations inside the filtrate. And so that means that that slows down the production of filtrate so that the filtrate can be modified, right, to a greater extent, and the salt can be reabsorbed. Okay, so in your book, they actually have a flow chart of all of this that looks something like this. Okay. Okay, and this actually has a flow chart of the opposite of what we just talked about. So like the top one, if there's too little blood pressure, then there's going to be less stretch in the smooth muscle walls, so the walls are going to actually dilate, right? And then that's going to increase your GFR, right? If you look at the macula densa, if the filtrate flow decreases and there's lower salt concentration, then they will actually cause vasodilation, and that will cause GFR. So it works in the opposite direction too, right? And then we also have some other cells in the kidney, which are called the granular cells. And the granular cells are the ones that act through aldosterone and renin, which we've already talked about, by causing the kidney to release those substances. So another function, so just ignore that one right here. So this would be 3, right? We talked about macula densa. When we look at the hormonal control, this is by the granular cells. Let me make sure that's right. Granular, yeah. Granular cells. So they produce renin in response to low blood pressure. And then this ultimately causes vasoconstriction and the release of aldosterone. So this causes vasoconstriction. Two, it causes the release of aldosterone. And that increases blood volume. Right? And this, all of these function into increasing blood pressure. Okay, so that would be a hormonal kind of regulation. So the control of the flow of blood into the kidney has to be highly regulated to filter the blood of toxins, right? To do a good job of filtering. But if it's too much, then you could damage the membranes, and then you could have renal failure because of that. So people that have uncontrolled high blood pressure, sometimes they'll get renal failure, right? Because of the blood pressure is just so high that kind of blows out their filtration membranes. And then you kind of get one sign is you get blood in the urine. Okay, so you actually see red blood cells in the urine. Okay, so this shows also the nervous system. So the sympathetic nervous system, right, can cause the release of epinephrine, which can also boost systemic blood pressure. Okay, so this is a good overall diagram that shows the ways that blood pressure and filtration rate are regulated. Okay, so let's talk about urine formation. So there are steps. We just talked about filtration formation. The second step is called reabsorption. Okay. So this is where you take stuff from the filtrate. So if I'm looking at the filtrate and the blood, right, I'm taking it, here it goes from the blood to the filtrate, right? So substances move from the blood into the filtrate. Here they move back from the filtrate into the blood. So that would be reabsorption. And that is primarily when we look at nutrients, that would be like glucose, right? That would be like amino acids, right? We have salt, like sodium and chloride, which we just talked about. We can also reabsorb ions. So you could actually reabsorb hydrogen ions. You could reabsorb potassium ions, right? So all of those things that might be necessary are taken back up, okay? Now, one of the signs, cardinal signs of diabetes is that you have too much glucose in your urine, right? So you have glucose in the urine. So if we look at this, a sign of diabetes is glucose in the urine. Right? And the reason why that is if blood glucose levels are too high, there are not enough proteins to transport the glucose back into the blood. Too high. There are not enough proteins to transport right? So you get, if you have high blood glucose, it gets into the kidneys and you're like, your kidneys are like, well, we need to get that glucose back into the body, right? Because we need glucose, we need to conserve glucose. But in order to do that, you have to have proteins that are channels that allow the glucose to get back into the blood. And if you have such high blood glucose levels that you don't have enough proteins, then you're going to end up having glucose in your urine. And so that's one of the tests for diabetes, is to test to see if there's glucose in the urine. Okay. Most of the reabsorption, like 65% of the reabsorption, takes place in the PCT. So that's the proximal convoluted tubule. Some of that reabsorption takes energy, some of it is passive. So if we look at a diagram that shows that reabsorption in your book, okay. So this is, the blue is showing reabsorption. So notice how stuff comes out, salt, bicarbonate ions, hydrogen ions, glucose, amino acids, and other nutrients. Even water has to be reabsorbed, right? And then we also have some reabsorption taking place, a little bit of reabsorption taking place in the DCT and also in the collecting duct. But most of it, 65% of it, takes place in the PCT, okay? Now this green part is the opposite of reabsorption. It is actually called secretion. Oops. Okay, so the third process is secretion. So this is where stuff that didn't get put into the filtrate will then be put into the filtrate. So this goes from the blood back to the filtrate. So it's secreted into the filtrate. So it's the opposite direction. It's the opposite of reabsorption. Okay. An important one would be like hydrogen ions. So let's say if the blood was too acidic, right? We can actually secrete hydrogen ions into the urine, and then our urine becomes really acidic. But it's a way to get rid of excess hydrogen ions in the blood, right? Some drugs also, potassium, and then some drugs are also actually secreted into, we'll put some drugs. I think penicillin is one of those. It's actually secreted into the filtrate. Okay, way to get rid of it. Okay, so if we look at an overall diagram of this, right? This shows reabsorption, actually filtration. This shows reabsorption, and then this shows secretion. And so all of this is the function of the peritubular capillaries, right? So the peritubular capillaries surround the tubules, and they allow for substances to be picked back up, reabsorbed, or substances to be put back in or secreted into the back into the nephron, the tubules. Okay, we already talked about reabsorption. Okay, so let's talk about the loop of Henle. So the loop of Henle actually has a descending limb. So this is closest to the proximal convoluted tubule. So this is the part of the loop of Henle that goes down into the medulla, and the filtrate goes through it in one direction, okay? This one is permeable to water. Not permeable to salt. So what this means is that water is reabsorbed. So it actually moves out due to the difference in the solute concentrations in the interstitial fluid of the medulla. As but found in medulla. So think medulla, all the fluid that is surrounding the cells in the medulla have lots of solutes in them, so it just sucks it out. And so this is an example of osmosis. And remember, one of the cool things about osmosis is that it does not take energy. Right? So it takes no energy. And so it's an efficient way to just suck the water back out of the filtrate and reabsorb it. So we don't want to have to end up pumping water. That would take a lot of energy to pump water back into our blood from the filtrate, so we just suck it back up. Okay? So if we look at a diagram that shows this, this actually shows the solute concentrations in the medulla. So as you go down, you notice that specifically salt and urea and other solute concentrations get progressively large, right? So this is very concentrated and this is not so concentrated, right? And so what this means is that as water moves out, you know, it's changing the solute concentration inside, but it's still sucking more out because of the difference, right? So it's a really efficient way of getting the water to leave, right? So even if solute concentration is really high down here in the tubule, because it's so high out here, water will even leave down there, right? And it'll actually make it more concentrated, okay? So that is as opposed to the ascending limb. So this is permeable to salt, not to water. So salt gets pumped out. And this takes energy. But this is important because this regulates solute concentrations in the medulla. So that pumping of the salt back out helps to maintain the difference in the solute concentration high up versus low down. Okay. So if we look at this, this is a diagram from your book. Okay. This shows the descending limb, right? Water gets drawn out and it's passive. So blue is passive, does not take energy. In this case, the ascending limb, water gets stuck in there. It cannot move because that would be important because we don't want water to move back in, right, into here because this might be more concentrated, the solutes, so it would suck water back in. So it's really important that the ascending limb is not soluble to water. And so salt gets pumped out. And then that sets up this concentration difference in the medulla. It's kind of, they call it a positive feedback mechanism that helps to maintain that difference. So this would only be really important in the juxtamedullary nephrons, where the nephrons extend down into the medulla. Okay. So this is an overview of the diagram. And so the whole function of the loop of Henle, right, is to reabsorb water and to help to concentrate the solutes, right? So we concentrate the solutes in the filtrate. Okay, so I'm going to stop there for today, and then we will talk more about this next week. So our next quiz is not till next Wednesday. Yes.
I really actually like most of those. Because are sometimes when I don't quite understand it, and I read it, and I listen to it.
Yeah. I used to tell students, as long as I don't have to listen to it, because I don't like the sound of it.
I understand. Whenever I play videos. Yeah, the time that I go over it, I'll go over my notes and I go over that.